For the next, our presentation is Analysis of Piperazine Derivatives, presented by Daniyor, Narmetov, Tulkun, Zhonovich, from Kazakh National Medical University, named after HD Asfendiyarov.
Good morning, dear colleagues and honorable judges. Today I'd like to present you my research titled In Silico Study of New Piperazine Derivatives. Nowadays, drug development faces a lot of challenges, such as low efficacy, expensive costs, unpredictable adverse effects, and lastly, long duration. With the help of in silico methods, those problems can be partially solved. Firstly, talking about low efficacy, it can be solved by early dropout of non-promising drugs. That will significantly increase the efficacy, and because we will spend less resources on them, we will have lower expenses furtherly. After it, talking about unpredictable adverse effects, all interactions with our organism will be studied, investigated during preclinical studies, so we'll have safer research. And talking about long duration, we will have significantly faster preclinical studies that will shorten the study. Here you see the Viagra drug development. It took about 10 years, and at the beginning they had over 200 molecule candidates. Only one of them achieved success in the end, and totally it took about 10 years and $750 million. In 2018, there was a research in Q1 journal, JABA. It was about drug development cost, and they found out that average drug development takes about $1 billion or sometimes even more. The goal of our presentation was to investigate new piperazine derivatives via computer modeling systems. We had two objectives. Firstly, to predict pharmacokinetic, pharmacodynamic, and physicochemical properties, and secondly, to find out possible targets and check mechanism of interaction with them. Talking about materials, firstly, you see 10 new piperazine derivatives, new drugs, and we put them to Swiss ADME tool. Swiss ADME is giving us a pharmacokinetic profile of each drug. Talking about ADME, it stands for absorption, distribution, metabolism, and excretion. PAS tool is giving us information about pharmacodynamic screening, with which proteins it's actually able to react. And according to the data we obtained here, we found out that our drugs are mostly effective in inhibiting following proteins: NMDA receptor and calcium channel. After it, we put them to the autodoc vina, or the program for docking process, and used PyMol for visualization of the united drugs and proteins. Discussing results, let's start with Boyle-Tech model. It was suggested by Swiss ADME, and here you can see two main parameters. Firstly, is if point is located within the yellow field, then the drug is able to pass through blood-brain barrier. If it's in white part, it's not able. If the point is blue in color, it's able to be a PGP substrate. If it's red in color, it's not able to react with P-glycoprotein. Only 4 drugs have been considered as able to pass through BBB. Inhibition of cytochrome enzymes. 3 drugs are not able to inhibit any of cytochrome enzymes, and 253rd one is able to inhibit all 5 out of 5 P450 enzymes that will lead to more toxicity for us. Nextly, skin permission, the highest one in 253. Later on in vivo studies, we found out that if you use it topically or right on skin, it will cause ulceration on skin. Lipophilicity and molecular weight. Lipophilicity 253, highest one, and lowest 268. Highest molecular weight also 253, and lowest 242, 67, and 68. 3 drugs have been considered as poorly soluble, and 7 remaining drugs have been considered as moderately soluble. Discussing pharmacokinetics, I'd like to say that all of them have been well absorbed in gastrointestinal tract. All of them are reactive with P-glycoprotein. Bioavailability score is 0.55. It means that about 55% of them is being absorbed via the gastrointestinal tract. 4 of them are able to pass through blood-brain barrier. Drug likeness is a parameter that's demonstrating how similar the drug with already existing drugs, and you see that 7 of them have passed all 5 filters out of 5, and 253 has the worst result with only 3 filters out of 5. And pass gave us information that these proteins are able to interact with following drugs. Here we see the comparison of binding affinity. For calcium channel blockers, inhibitors, we chose nifedipine as the gold standard, and among other drugs, among our drugs, I would like to highlight the 253 because of their highest binding affinity. And among NMDA, there is a ketamine as a gold standard, and 276 as I would like to highlight it because of best result. Here you see the NMDA receptor and its active site. Here, on the left side, you could see the molecular formula of the 269 drug interactions it's getting with this protein, binding affinity, and also the exact reaction with which amino acids our drug is interacting with which type of interactions. 270, 71, they have similar binding affinity because of similar structure, and 76 was the highest binding affinity. Here you see the calcium channel with verapamil, binding affinity, highest one in 253, 67, 71, and conclusion. To conclude, I'd like to say that the first computational study of new piperazine derivatives, it's novel drugs which are produced in our university, and this study has validated them as able to target therapeutically in further therapeutic use for targeting NMDA and calcium channels. That's all. Thank you for your attention.
So, to the very beginning of your presentation, what is the rationale of each compound that you synthesize, and yeah, how can you come with these list of molecules at the beginning?
You mean this?
Yes.
Oh, could you please repeat the question?
How could you ending up with these 10 compounds, and to compare with the original medication piperazine?
As I understood, you mean why I'm comparing those drugs with nifedipine and ketamine, right? Ah, okay. We're comparing them because they have the same therapeutic use. Because here in PASS we obtained, according to this data we obtained here, we understood that our drugs are able to inhibit those receptors. And ketamine, for example, is also an NMDA inhibitor. Calcium channel nifedipine and verapamil are considered as calcium channel inhibitor. That's why we are comparing them. That's an answer.
Can you go to the very first one of the 10 compounds? I mean, how could you end up with this list of 10 compounds? So, I mean, where is the action site of this compound to compare with the original one? Why end up with these 10, not the other 10 lists of the others?
Because for our study it was produced in our university. Like there were a lot of other drugs, of course, but for research it was only those ours. And why? Yes, if you ask about why exactly piperazines, because they have broad effect clinically, they are widely used in as anxiolytics, as antidepressants, as buspirone, risperidone, antipsychotics. Also, piperazine citrate is widely used as antihelmintic. So, and also because of their piperazine ring, they are robust in their structure, so that's why they are good candidates for future possible drug.
Is this all compound is newly synthesized, or
Sorry?
is this all compound are newly synthesized, or is already published?
It's already synthesized, yeah.
It's already synthesized, and have any body report this action before, like
No, it's the
as
first study of their interaction with human body.
okay.
And
So
these drugs have...
For this or derivative, is there any report with
No.
others biological action? No.
No, those drugs have been produced in our university like recently, and the novelty of the drug of the study is that it's the first study to analyze their pharmacokinetics, pharmacodynamics.
All right. Okay. I just have one short suggestion is that, so if you have the molecules that not really is published the action yet, in general, it's gonna be good for you if you do not show the structures, no? Because it's gonna be very safety for you. And if you have already sure the actions, biological properties, and published, or you patented already, then you can expose all the structures. That's just
Sorry. Okay, understood you. Thank
little
you.
suggestion. That's
Okay, thank you so much.
Well, thank